Thank you all so much for joining us this morning for International Elephant Foundation's Conservation Chat. We just really appreciate you all being on here with us, and we have a great speaker lined up this morning. I want to go over a couple of housekeeping items before we go further. If everybody could please put their microphones on mute, that would really be helpful. Sometimes it can get a little distracting for when we're having the speaker. So everybody could put their microphones to mute. And then once the speaker starts, we'll ask that everybody shuts off their cameras so we can save bandwidth. So that would be good. And then as we go through the presentation this morning, if you have any questions, we ask you to put them in the chat. And then we will, towards the end, if we have more time, we might be able to let people unmute and ask questions directly to
Hello. Hello, how are you?
I'm good.
So yes, so with that, I'm going to introduce our conservation coordinator, Sarah Conley, and she is going to introduce our speaker. So take it away, Sarah.
Thanks, Julie. Hi, everyone. Good morning, good afternoon, wherever you are. Good evening. We are so lucky to have today's speaker today. Erin Latimer is someone that everyone in elephant management should know, and she has done so much to help people and help elephants that it's really a treat that she gets to share her expertise with us. So she's a research biologist at the Smithsonian's National Zoo Conservation Biology Institute and is the laboratory manager for the National Elephant Endotheliotropic Herpesvirus Laboratory, or EEHV. Elephant endotheliotropic herpesvirus is the leading cause of death in young Asian elephants in human care, but it's also been seen as a cause of death for African elephants in human care and in the wild. So it is a huge problem when it comes to protecting and continuing elephants for the next generation. And Erin is on the cutting edge of dealing with that. She's also the founder and executive committee member of the North American EEHV Advisory Group and member of the Asian EEHV Working Group and the African EEHV Working Group. Capacity building has been a major emphasis of the EEHV Lab that she's in charge of. She has helped train staff, equip EEHV laboratories in range countries in Africa and Asia, as well as in North America and Europe. She is an expert on these things, and we are so lucky to learn from her today. So please, if you have questions, put them in the chat. There's a lot to know. I'm looking forward to learning stuff today. So thank you, Erin, for joining us. Welcome.
Well, thank you. I'm happy to be here.
All right.
So today I'm going to talk about elephant endotheliotropic herpesvirus. But I want to start out with the acknowledgement first instead of at the end, because it's so important. IEF has been an incredible supporter of my lab over the years. They supported my salary for several years and then donated the qPCR machine since 2014 for the monitoring of the elephant calves. My lab wouldn't exist in its current form without the help of IEF. And then there's Debbie Olson. She's my go-to source for advice on everything to do with elephants. She knows everybody in the elephant community, and she is so good at breaking any issue down to what is really important. We are so lucky to have her help on EHV. So I'm so happy to be able to maybe help out IEF a little bit by giving this talk. So I'm going to give you the basics of elephant endotheliotropic herpesvirus, or EEHV, by going through each of the letters in the abbreviation. We'll start with the first letter, E for elephant. So why do we care about this virus? It's the leading cause of death of young Asian elephants in human care. On average, over the last 40 years, it has killed about 1 Asian elephant a year in the US and more worldwide. With only 40,000 estimated Asian elephants globally, each death, especially in the young reproductive age group, is tragic, and preventing these deaths is vital. It was first identified here in 1995 at the National Zoo after Kumari, a 16-month-old elephant calf, died after a brief illness. A necropsy was done by the veterinary pathologists. Hemorrhages were found throughout the calf, and evidence of a viral illness was seen. A retrospective search of previous unexplained calf deaths uncovered an additional 9 cases globally going back to 1980. Since then, over 130 deaths in Asian elephants have been identified, both in human care and in the wild. EHV predominantly affects younger elephants. In Asian elephants, it's usually in the 1 to 8 year range, and it skews a little older for the African elephants, up to about 15 years old. Calves are protected by maternal antibodies until they are about 2 years old. Once the maternal antibodies start to wane, they are at increased risk of getting EHV. If they are exposed to EHV before the maternal antibodies wane, they can mount their own protective immune response and are protected from serious disease. So before 2019, EEHV was predominantly an Asian elephant disease and had only been seen a handful of times in African elephants. As of 2019, we are seeing it increasingly in African elephants in human care. Since 2019, there have been 4 deaths and 17 illnesses in African elephants in the US. We don't know why there's been such a dramatic increase in EHV disease in African elephants in these last few years. In addition to the increase in African elephant death and disease, in the last 2 years, we have lost 5 young Asian elephants just in the US. Rather than the average of 1 death per year that we have been seeing, we lost Batu and Ajay at the Rosamond Gifford Zoo in New York, Jasmine and Thorn in Albuquerque, and last summer, Biko at the Columbus Zoo. These deaths in the reproductive age elephants are especially difficult for the elephant population as a whole and for their caretakers and the zoo-going public. So the second E stands for endotheliotropic, which means that the virus targets and reproduces in the endothelial cells that line the blood vessels. As the virus reproduces, the endothelial cells lyse, which causes the hemorrhaging, and the edema, which are hallmarks of the disease. With it being a hemorrhagic virus, you can think of EHV as the Ebola of the elephant world. So we'll move on to the H, which is for herpes. EHV is somewhat related to the herpes viruses that humans get, like herpes simplex, chickenpox, Epstein-Barr, which causes mono, and Kaposi sarcoma virus in AIDS patients. Like other herpes viruses, it is species specific, so it only infects elephants. As humans, we are not going to get EHV, and the elephants are not going to get herpes simplex or chickenpox from us. Like the human herpes viruses, these elephant viruses go latent in the elephants after their primary or first infection, and virus may be shed in their trunk secretions at a later time. This is probably how other at-risk elephants are exposed. This is similar to how humans can pass herpes simplex from a cold sore when latent herpes simplex reproduces or comes out of latency. EHV has coexisted with its elephant hosts for millions of years, just as other herpes viruses have coexisted with their hosts. So contrary to what the animal rights people sometimes say, EHV is not a disease of captivity. So the last letter, V, is for virus. EHV is a double-stranded DNA virus. The important thing about this is that it doesn't mutate rapidly, unlike SARS-CoV-2, HIV, and influenza. They all mutate quickly because they are all RNA viruses, and this is why we need a new flu vaccine every year and why there have been so many SARS-CoV-2 variants. As a double-stranded DNA virus, EHV mutates very slowly, so an eventual vaccine should be effective for a long time. So there are 7 main types of EHV. We have EHV 1, 4, and 5 in Asian elephants, and EHV 2, 3, 6, and 7 in African elephants. EHV 1 in the Asian elephants causes by far the most morbidity and mortality. We don't yet know why there is such a difference in the pathogenicity and prevalence of these different types. So EEHV is the name of the virus. EEHV hemorrhagic disease, or EEHV-HD, is the name of the serious disease that it causes. Herpes viruses are not supposed to kill their hosts. Chickenpox and herpes simplex cause disease but don't kill healthy humans. For some reason, the EEHV viremia in young elephants sometimes gets out of control and leads to EEHV hemorrhagic disease, which is often deadly. Elephant calves receive maternal antibodies from their mothers. The antibodies have been shown to wane by about 2 to 3 years of age. We believe that if they are exposed to EEHV while they still have maternal antibodies, they are able to mount their own immune response and recover. If they are not exposed at that young age, the EEHV viremia can get out of control and lead to EEHV-HD. The human corollary is chickenpox, which is usually a mild disease when young, but can be dangerous to get as an adult. So how do we detect EEHV? The gold standard for diagnosis of EEHV is quantitative polymerase chain reaction, or qPCR. This is the same type of test that they are using when they talk about the COVID PCR test. It's a way of amplifying small pieces of target viral DNA so that we can see it and quantitate it. The zoo veterinarians are able to use the information about the viral levels in the calf's blood to guide treatment and determine prognosis. The current treatment aims to decrease viral replication, reduce damage caused by the virus, and give supportive care. Human herpes antivirals are used, but we are not sure if they are effective. What does seem to be most effective is supportive care, along with plasma and platelets, which treat the hemorrhage. Each case of HD has led to more insight on what are effective therapies for EEHV-HD. Stem cells and treatments that support the innate immune response have shown recent promise. Paul Lang at Baylor College of Medicine has developed serology tests which measure the level of antibodies a calf has, which can tell us which calves are at risk of developing EEHV-HD. He is also developing a vaccine for EEHV-1A, which may be available in the next few years. Until the vaccine is available, we depend on weekly or twice weekly monitoring of young elephants to detect the virus early, especially in the calves that are at higher risk because of a lack of antibodies. So now I'm going to talk about what we do here in the lab at NZCBI. We know that low levels of EEHV can be detected in the blood of young elephants before clinical signs appear. We also know that early aggressive treatment leads to increased survival. This means that it is important to routinely monitor the young elephants to detect the early stages of the viremia. Virtually all of the U.S. elephants under age 15 are tested at least weekly for EEHV. Most of them were tested in the lab here at NZCBI using PCR on whole blood. We can detect low levels of their viral DNA and give the vets a heads up that the calf may end up with EEHV-HD. This will trigger increased monitoring of the calf and allows for the prompt aggressive treatment, which has been shown to increase survival. The EEHV monitoring of the U.S. elephants that we do here at NZCBI is set up like a nonprofit business. The U.S. zoos pay an annual membership fee to the lab based on the number of elephants they have and the type of testing they need. It covers their routine testing, as well as the increased testing of the calf and herd that is needed when they have an EEHV-HD case. These fees cover the lab supplies and equipment, as well as the salaries for the lab staff. We test samples from almost 30 U.S. zoos every week. Each year, we test about 7,000 elephant samples total. One of the most rewarding aspects of the job is the capacity building that we do for the global elephant community. We have helped to equip and train EEHV labs in numerous range countries, as well as in Europe and North America. Many of the people that we have trained have gone on to train additional people in their countries and expanded the EEHV testing capability worldwide. This molecular diagnostics training can then also be used by our colleagues to look for other pathogens in other wildlife species. We also have a mobile lab so that when a zoo in the US has an EEHV-HD case, we can send a technician and equipment to the zoo and test on site. providing same-day results for the duration of the case. The EEHV Lab collaborates on research projects with colleagues around the U.S. and internationally. This research includes projects looking at biomarkers of serious disease that might lead to earlier and improved diagnosis and treatments, as well as investigating the genomics of which elephants will get serious EEHV disease. Funding for the lab itself and the research projects have come from a variety of sources, mostly US-based. We contribute to a few peer-reviewed scientific articles a year. Here are the papers published in the last 2 years. In them, we describe the types of EHV found in wild elephants in South Africa, reported on an unusual subtype of EHV1 that killed 3 orphan calves in Sabah, Malaysia, reported on changes in biomarkers during EEHV-HD disease in 2 different studies in the US, contributed to Dr. Lin's work on EHV serology, and described several of the recent EHV cases in African elephants at the NDC. In 2013, we helped organize what was known as the EHV Advisory Group. It consisted of about 25 vets, researchers, elephant husbandry experts, and communications people, and its aim was to provide help to the elephant community when dealing with EHV. Since then, we have helped to establish 4 region-specific advisory or working groups globally that work together closely. We provide information on EHV monitoring, treatment, and research, organize research symposiums every year or 2, often in conjunction with IEF, facilitate research collaborations, and provide capacity building. One of the things the North American Advisory Group has done is host a website, the ehvinfo.org. We provide information on the 4 advisory and working groups, a list of EHV publications, and recommendations on monitoring, translocations, treatment, sedation, and necropsies. We also provide contact information for the EHV testing labs around the world. So I'm happy to answer any questions you have now or anytime in the future. You can just email me.
Thank you, Erin. That was great. I think for anyone that doesn't know about EHV, that was a great way to sort of give people a quick up to speed. So I really appreciate that. So why don't we talk about why should the public care about EHV? Why is this a problem that people outside of the elephant management community should be focused on?
Well, because it's the leading cause of death in young Asian elephants in human care. We're also seeing it in the wild. So in addition to the ones that we're taking care of in human care, we think that it's going to increasingly become a problem in the wild too, as there's habitat loss. The larger herds that have been seen in the past are going to become smaller herds. They're not going to be mixing. And we think that they won't get exposed to EHV at the young age that they should. So we don't know a lot about it in the wild yet. As we are helping to set up labs in other countries, we're learning more about what's in the wild. But we think that it's going to become increasingly important in the wild too.
That makes sense. Someone in the chat is asking, when it comes to EHV, what is considered a young elephant? So what's about the age range where this is a factor?
Okay, so for Asian elephants, it tends to be about what we used to think was about 1 to 6 years of age. But now in the last couple of years, there have been some older ones. So for Asian elephants, it's more like 1 to 9 years of age. So just those young elephants that are soon to become reproductive. It's just such a tragedy when we lose them. And for African elephants, it's a little bit older. It tends to be more like 6 to 15 years of age, but there have been some younger ones too. We don't really know why there's the age difference, but they are separate viruses and separate elephant species, so there are differences.
Okay. And I know we've talked about, that EHV can turn into EHV hemorrhagic disease, but can you talk about what specifically happens to the elephants? Because I think I know as humans, when we think herpes, we think skin rashes, we think things like chicken pox, things like that. And that's not what happens with elephants when they get EHV-HD. Can you tell us about that?
I will try. I'm not a veterinarian, but I have listened to the talk, so I will do my best. So they get a viremia, or they get this virus in their blood. So it's not like herpes simplex, which is a cold sore on the surface. You know, it's a viremia, it's in the blood. And we see it becoming, it ends up at very high levels in the blood, and it starts killing the blood cells. There's a monocytopenia, which means the monocytes are being killed. A leucus, the white blood cells are also being killed, and the platelets. A vet would be able to explain this better, I'm sorry. But anyway, it's, and it attacks the endothelial cells, which line the blood vessels. So as it's reproducing in those vessels, reproducing in the cells that line the blood vessels, those cells lyse when they release the virus. So then the blood vessels become leaky, and fluid comes out of the blood vessels, and you see that edema that was shown in one of the pictures. The elephants look swollen. We also see hemorrhaging in the eyes and in the tongues. And then if it's an unfortunate case where the calf dies and they open it up, there's usually a lot of hemorrhaging in the heart, the liver, the intestines, and the spleen. Any organ that has a lot of vascularity, they'll see hemorrhaging in. So it's just a devastating disease for these poor elephants.
Okay, so to put that maybe in layman's terms, that's their blood vessels are bursting, they're bleeding internally, they're swelling significantly. It's incredibly uncomfortable and very difficult to treat.
Yes, they're making great strides in treatment, but still, even if we can catch it early, they're not always able to save every calf.
So that brings up an interesting thing. You talked a lot about how the best line of defense is early detection, which you can tell by viral levels in the blood way before you see clinical symptoms. And why is that important? Because I think maybe people outside the elephant world might not realize that as soon as you see clinical symptoms, your baby elephant might be too far gone.
Yes, so what we have seen is if the elephants aren't being monitored and the first indication that there's an EHV viremia going on is clinical signs, it's almost always too late. So what we need to do is find it early. So Ramiro Essalza at the University of Florida Vet School did a great study. He looked at the survival rates with early treatment, or later treatment after clinical signs are seen, or no treatment at all. And with no treatment at all or late treatment, which is with significant clinical signs being seen, the fatality rate could be like 80%. But when it was caught early and they gave them really good supportive care, stem cells, that kind of stuff, the plasma and platelets, fatality rate went down to 25%. So, big difference in getting early aggressive treatment to these calves. And we do that by monitoring. We test them every week or twice a week, and we can detect as little as, 100 or 200 viral units per mil, which is not much. In a EHV-HD case that is serious, they can have up to 1 million or more, many million viral units per mil. So we can detect it really early, down at, just a couple hundred per mil. And we can give the vets a heads up. We can give the zoo a heads up that they're going to need to not only monitor more, but get ready for a possible case. They're going to mobilize plasma, platelets, all the supportive care. It usually takes around the clock care when they have an HD case. So everybody is, on duty, people are spending the night at the zoo. So we can just give them a little heads up that all of this is coming.
Wow, that's an invaluable tool. Because in an emergency, the more prepared you are, the better. So we have some questions in the chat. So dovetailing with the early detection, let's talk a little bit about what you know about the vaccine. If it's developed for one strain, how much protection might it provide against the other strains? I know that's not what you're working on, but do you have any thoughts or insights in that?
A little bit. We think that there might be some cross protection between some of the viruses. So like EEHV-1A, which causes most of the death and disease in the Asian elephants, might have some cross reaction with EEHV-1B. So it's possible that an EEHV-1A vaccine will help protect against EEHV-1B too. We don't know how much cross reactivity there is with any of the other ones. That's what I can say.
Fair enough. Yeah, that we won't know until the science tells us, right? So, other questions. What projects or studies are being done to evaluate EHV hemorrhagic disease prevalence in wild populations?
We are trying to help facilitate those by doing the capacity building. I know that there's a lot of labs now in Asia, and we're just getting kind of started in Africa. So Asia, there's, I think last count was maybe 9 labs in Thailand. So they're doing a lot of work on EHV in Thailand. They don't have as many elephants in the wild in Thailand. I think most of them are working, but they are doing projects there. There are several labs now in India. There's one in Nepal, Sumatra. We're working on Myanmar. Malaysia has a lab. So all of those countries are now in position to be able to test their own elephants to see what the prevalence is. We do need a lot more information about the prevalence in these range countries. And because it just has become an issue in African elephants since 2019, we're just getting started in Africa. So we've helped get a lab set up in South Africa. So we know a little bit about the prevalence in the wild there. The paper just came out. We are helping to get stuff going in Zambia and Zimbabwe. But unfortunately, the pandemic kind of made it more difficult. We're hoping to do some work in Uganda. And all of these labs that we help set up will be able to get an idea of what the prevalence is in the wild. We do know that wild elephants in Asia and Africa do have this virus because it's been with the elephants for millions of years. It's been coexisting with them. We just don't know the extent that it causes disease, the hemorrhagic disease in the wild. But we do know it's there naturally.
That's good to know. So maybe just for our audience, why is it so challenging, besides lab capacity, to get good data on wild populations for this?
Well, right now, the best sample is blood, and that's pretty tough to get from a wild elephant. We can also determine prevalence by using the trunk washes, which is a way to measure their trunk secretions. So if an elephant is being immobilized for something else, for translocation, they may be able to get trunk washes or blood or swabs. There are a few groups that are working on detecting it in fecal samples, which would be the best way because it's non-invasive. But right now, the science isn't there for how best to detect it in the fecal samples. It has been detected, but it's not as good of a method as like a trunk wash or blood.
That makes sense. So speaking of trunk washes, for our audience, can you say what they are? And then we have a question about trunk washes.
Yes. So a trunk wash originally was developed as a way to test for TB. And what it is they have trained elephants to suck up about 50 mLs, which is a couple of ounces of sterile saline. They suck it up into their trunk, and then they're trained to lift up their trunk so that the saline, runs down the inside of the trunk. And then they're trained to spit it back out into a Ziploc bag. And then we can take that saline, which has, cells from lining the inside of the trunk. It may or may not have the EEHV virus if they're shedding it. And it also has lots of hay and mud and dust and stuff. But we are able to take those samples and look for EEHV virus. And we can detect shedding of EEHV in these samples, especially from African elephants. They seem to shed more. Asian elephants don't seem to shed as often. But we can detect when they're shedding. It doesn't mean that they have the hemorrhagic disease. It just means that they are shedding in their snot, their trunk secretions. And what we have seen is, we think that's the way that they expose the at-risk elephants. They're shedding it in their trunk secretions, they're touching everything with their trunks, so they can expose the calves. And if they expose the calves young, we think that's a good thing, because then the calves can produce their own immune response to it. But if we have a calf that's older, that has lost its maternal antibodies, and it gets exposed at that point, might not be such a good thing, because then it can get the hemorrhagic disease.
Okay, that makes sense. And that actually segues right into this question. Someone's asking, are there facilities that see shedding in the adults' trunk washes that later see active cases in the calves?
Yes, definitely. Especially now that there's so many zoos that are able to do testing in the US, we have more testing capacity, and a lot of the zoos are now collecting and banking trunk washes every week from the entire herd. And then if we have an HV case in a calf, we can then go back and look at all of those banked trunk washes. And what we're seeing is maybe an adult has been shedding, and then about a month later, we might see an EEHV HD case in a calf. So we're starting to figure out with all of this testing what the shedding looks like. We're hopefully going to be able to figure out how much shedding is necessary to expose a calf. And then after a calf recovers from an HV case, they then shed quite a bit of EHV in their trunk secretions out into the environment too.
Wow. Okay. And follow up from the person asking, do you know how many facilities are doing this work or have seen this connection?
I would say most of the breeding facilities in the US are either now banking trunk washes or planning on it soon.
That's amazing how just one sort of indication to help us learn, everyone hops to it and is getting the data and getting the samples out there so that we can learn more. That's wonderful.
Yeah, it's very labor intensive, but I think we're going to get a lot of really cool data from it and figure out kind of, transmission, what's needed, how long it takes. I think we're going to get a lot of good data from this.
Well, that's hopeful. That's great. Why don't we talk more about EEHV globally, the capacity building that you do? What is involved in that? Because situations, building a lab here in the United States is not quite like setting something up in Zimbabwe. What do you have to look for? What's involved in that? How does that work?
Okay, so what used to happen was somebody from another country, a range country, would contact me and say they wanted to send me samples to test because they had an elephant die. But by the time we get the samples, it's months later because CITES permits are needed. So what we started doing, I guess 10, 15 years ago was rather than having them send samples here, we would rather help set up the testing in whatever country it is. So what I try to do is if they are able to connect with a local university or a government lab that has at least lab facilities, or ideally that has qPCR equipment. We try to have them collaborate with the local lab because they will already have the infrastructure, the equipment, and possibly even technicians. And all we would need to do is provide protocols and reagents and training. So that's, I think, the way it works the best. It's a little cheaper than having to do everything from the beginning. And I think it's more sustainable. But we can, if they don't have a local group that they can hook up with, we can start from the beginning. And actually the first lab that we helped set up was in India. And it was, started from the beginning. A young forest vet in Kerala, India, had a building that he got some money to refurbish as a lab. And we helped supply him with some of the basic equipment for conventional PCR, which is a little bit cheaper. We went over and trained him. We brought all the reagents that we needed. And then we kept in touch, with troubleshooting and stuff afterwards. So that's what we do. We figure out what's needed, what's the best way to provide what's needed, and then provide the training so that they can do it themselves.
That's great. That's very important. And that's important because number one, the samples need to be run fairly quickly. Yes, it's not like you can wait for them to be shipped internationally. And then number two, there are global rules about shipping these types of things internationally, correct?
Yes. Yeah, definitely. If an elephant was sick, we would not be able to get the samples in time internationally. It was only like if an elephant had already died, they would try to send samples. But this way they can test, right when they need it, when the elephant's sick, so they have a chance to treat the elephant and, it'll survive. And then they have the lab capacity to continue on to test their EHV, do their research projects, look at the prevalence in the wild. And they can also use the same equipment and the same training to look at other diseases too in other animals. So like the first lab that we helped, we just went over with the equipment with the intent of helping him test for EHV. But then he was able to kind of leverage that to start publishing papers and got more money from his government, and then ended up with a full-fledged wildlife diseases lab.
That's incredible. That's fantastic. Some more questions in the chat. Going back to trunk washes. When submitting trunk washes, do you prefer to have the submitter centrifuge the sample, obtain a sediment/pellet and decant the supernatant, or do you prefer the sample in its entirety?
Actually prefer it to be centrifuged than pelleted. It just is a little easier on us. Plus, we think that the EEHV virus is in the cells. And if those cells lyse, they're going to release the viral DNA into the liquid, which when we get it here, we centrifuge and pour out the liquid. So if we can get it centrifuged and processed at the zoo, just the beginning centrifugation and pelleting, I feel like we're not going to lose any viral DNA that might be there.
Great. The commenter says perfect, thank you. So I think that'll help the data collection right there. So that's great. So you are doing all this work with elephants in human care because that's how we get samples, and that for now. How do these partnerships with elephants in human care and facilities, how does all of this help with these efforts?
Because these are the samples we can get, like you said. Getting samples from the wild is a lot more difficult, more expensive, and almost impossible unless you're going to be able to immobilize the elephants. So these elephants here in the US are trained for vet care and for husbandry We can get the samples, with very or little or no stress because they're trained for this. They're almost always happy to comply because they're getting treats. So we can get all of these samples and figure out what's going on, which if we had to depend on just getting wild samples, we couldn't do. But all the stuff that we're learning, we can then apply to animals in the wild.
Okay. I mean, obviously this is all conjecture because we're still learning about the disease and figuring out the vaccine and things like that. But how do you envision this work being used in the wild?
Well, I guess, I don't know a lot about the vaccine, but if it could eventually be given to elephants in the wild, and we can prevent in the wild what's going on here, that would be great. I don't know how that would look. I'm not a vaccine person or like an elephant conservationist, so I don't know how that would work, but it would be great, yes, if we could prevent the same kind of thing happening in the wild that is happening in human care.
Great. Yeah, that would be amazing. What do you think people should know about EHV in general? Like, so if they walk away from this talk today, what do they need to carry with them? What should they share? If someone asks them how their day was and they talk about this amazing thing they learned, what should they share with some stranger or their coworker?
Ooh, good question. I guess I think what's amazing is how much collaboration there is in the elephant community. When I first started this job in 2004, we really didn't know a whole lot about EHV. But the elephant community is so collaborative and is willing to work together and give any information to anybody that needs it. They're willing to collect samples and do whatever's needed to try to learn more about this disease. And I mean, we have made amazing strides and amazing increase in knowledge about it. When we first started, we thought that if we saw a little bit of EHV in the blood of an elephant calf, that it was going to die. But now we know we want to see it when they're young, when they still have maternal antibodies, so that it can be controlled, the latent, and that elephant calf can produce its own antibodies. So, it's not a death sentence when we see these low levels. But we know that because everybody has worked together to collect samples, to share knowledge, and to help everybody out. So it's just been amazing how much collaboration there's been with this disease.
That is amazing and really a testament to the elephant community. I meant to ask you, so this year you are co-principal investigator on a project that IEF is funding. Can you tell us about that?
I will try, but again, I'm not a vet, so hopefully you will have one of the vets involved doing a talk too.
Yes.
So we're looking at something called D-dimers in elephant calves, and we're looking at it in healthy elephant calves and in elephants that get EHV, either just a controlled viremia or hemorrhagic disease. And D-dimer is something that's involved in blood coagulation. And the coagulation cascade is really complicated, and I don't know how it works. I actually work on a different part of the cascade, something called protein C in humans. But it's a really complicated cascade, and D-dimer is one piece of it. So we're looking at levels of D-dimer in healthy and controlled and hemorrhagic disease, and hoping to see if we can use these levels to kind of figure out the prognosis of a case when we first get it. Because like when we first detect EHV in a calf, I let the vets know that yes, we have EHV1 or EHV5 or whatever in this calf. but we don't know which way it's going to go. If we know that the elephant has antibodies to that particular type, we can kind of guess and hope that it's going to be okay. And if we know that the calf is seronegative or doesn't have antibodies, we can, we'll be much more worried that it's going to go to HD. But we're looking at all these different things like D-dimers and troponin, and something called SAA and haptoglobin. And we're hoping that we can figure out like what happens with the levels of all these different biomarkers during the beginning of an EEHV viremia, so that we can kind of get an idea of, is this going to go bad or is this going to be a controlled viremia? So all the stuff that we can learn by looking at all these different biomarkers during these viremias can help us figure out, what's the prognosis going to be.
Yeah. Okay. More questions. So what brought you to work on EHV and elephants in general? How did you get involved with elephants? And then maybe touch on people like Gary Hayward and the work that they're doing, that kind of this work builds on the, I guess, EHV family tree, so to speak.
Okay. So I got started, years ago. I have an infectious disease and molecular biology background. And I started at the zoo. It was actually just a half-time hourly position that was grant funded after our elephant Kumari died. So I got involved, and I just really love, infectious diseases, and then just the environment with the elephant community. I really loved being able to help out. So I've just gotten more and more involved in it all, and have really enjoyed being able to help in the small way that I can. So from the beginning, Gary Hayward at Johns Hopkins has been involved. So what happened was, the disease was discovered here and described here at the National Zoo when Kumari died in 1995. And one of the veterinary pathology residents, Laura Richmond, is the one who described it. And then she finished up her residency here and went to Johns Hopkins to work with Dr. Gary Hayward to do more work on EHV. So she did a PhD with Dr. Gary Hayward on EHV. And he is involved because he is one of the human experts, or he is one of the experts on human herpes viruses. So he hosted her in the lab, and they developed the first PCR diagnostic tests for EHV, and Laura kind of really did a lot of the descriptive stuff of the disease, the early work on it. And then Dr. Paul Linning, who is now at Baylor College of Medicine, before he went to Baylor, he actually did his postdoc with Gary Hayward's wife at Hopkins on human herpes. Then he went to Baylor, and he got involved because his lab at Baylor is right across the street from the Houston Zoo. So when Houston Zoo lost their elephant, Mac, I think in 2008, Paul was like, well, I work on human herpes, maybe I can help out. So he got involved, and then has just been an incredible resource. So the two of those, Paul has developed the qPCR assays and is now developing the serology assays and the vaccines. And Dr. Gary Hayward is an expert on phylogeny, which is how the viruses are related. So he can look at a DNA sequence of this virus, and he's just a genius. He can look at sequence and know, okay, well, this virus is 5% diverged from this virus. These viruses have been coexisting with the elephants for millions of years because of the rate of divergence of these viruses. And he is the one who helped us, told us that there are all these different types of viruses. We first knew from Laura's work about EHV1 and 2, and EHV1 was in Asian elephants, EHV2 was in African elephants. But then as we started detecting more and more of them and sequencing them, Gary said, oh no, we now have EHV3. It's now 3A and 3B, EHV4, EHV5, EHV6, and EHV7. And he determines that it's a new virus by looking at the DNA sequences of them. And they're all related. They're all herpes viruses, and they're diverged by at most by like about 10% from each other. So there's many similarities between all these viruses, but there's also some differences. We do see a few differences in how they act in the elephant body. I think EHV3, we've seen a little more effect on the kidneys than like EHV1 has. But we're still learning a lot, especially like from Gary's work on, he can look at all these different genes from all these different viruses.
Yeah, amazing. I know from personal experience, listening to Gary talk about his work, you learn a lot, and then you're just, at some point, your brain's like, I can't keep going, because Gary he can see through the data and see things that the other people have a hard time following. So it's incredible what he's doing. So you touched a little bit on how his work helped us know that the virus co-evolved with elephants. Can you maybe explain that a little bit more?
Well, I think, when we first started looking at this, Laura found EHV1 in Asians and EHV2 in Africans. And the concern was that maybe the virus was being passed from one elephant species to the other, and that's why it was causing disease. Because herpes viruses can do that. So like humans, we get herpes simplex, and it's really just a mild disease. There are some non-human primates that get something called herpes B. And to them, it's a very mild disease. It's basically like our herpes simplex. But if we get herpes B from them, it's got a 70% fatality rate because it has crossed the species barrier. So that was kind of concern at the beginning. Was the problem that as we brought elephants into human care, they were getting exposed to viruses that they weren't supposed to get, and that's why it was fatal. But now we know, based on the work we did in India, seeing it in the wild, and Gary Hayward's work, that no, Asian elephants have EHV1, 4, and 5 endemic to them, and they have it in the wild. And it's the versions that they have in the wild are the same as that we have in here in human care. And it's not an issue that they got it from African elephants. He's able to look at the divergence of the different genes and know that they have been with the elephants for millions of years. I can't explain because I don't really understand, but they know how fast a DNA virus can change, just like RNA viruses mutate much more quickly. And by looking at the rate of change in DNA viruses and looking at the breadth of diversity of the different viruses that we have found in human care and back in India, he can determine that these viruses have been with the elephants for millions of years.
Wow. Okay. And just so everyone's clear, Gary's still working on this, right? There are still, yes. He's got so much more data to analyze, so much more kind of nuances and insights into these viruses that we still need to know about.
Yes, yes, he's still working hard. He's just amazing. He has so much data that still needs to be gone through. He's just been an incredible resource over the years. Nobody could do what he does. He's amazing. And he still has so much more to do. Right now, we're waiting for some data on the cases in Malaysia that he's going to analyze because it looks like the viral type that they got in Malaysia is diverged from the EHV1A that we have here and in the rest of Asia. And that may be because these cases on the island of Borneo, those elephants have been kind of isolated for hundreds of thousands of years. So maybe, these viruses that they have also been isolated, and that may be the cause of their divergence from the ones that we see in the rest of Asia and in the US. So he's going to be looking at that data soon. Yes, he's very busy.
Wow, crazy. Yeah, well, all of that is why IEF not only supports your work, but Gary's work and Dr. Ling's work. We're always looking for more people to support that work. We actively identified EHV as an important funding priority for IEF almost as soon as EHV was identified, and we're always trying to seek funders to look into and to support that kind of work. So it's really important, and we appreciate you talking about everyone's work, not just your own. That's really great. We do have a question in the chat about Colossal, which I know you aren't necessarily involved with them, but maybe you can speak a little bit about, because they claim that their work may help EHV research. So can you explain sort of how that work fits, or what your understanding is, or what you think about all of that?
Well, I know that they are helping to fund a postdoc that is going to be working on some of the vaccine work. So they have been, I think, very helpful in providing money for their different research projects. They're also going to help support our EHV advisory group meeting this summer. They're providing some support for that. So yeah, we always need money to help out, and apparently they've been able to raise quite a bit of money for their projects, and they are going to be using some of it to help out the EHV community.
That's great. And from what we know, if EHV, since it co-evolved with elephants, if Colossal's ultimate goal of de-extincting some form of mammoth, EHV will be a real concern. So they will need to address that, or at least it's in their best interest to address that.
Yeah, I would assume that if they did bring back another elephant species, it would also be vulnerable to the EHVs.
Yeah. Well, if I'm going to ask anyone, if you have last minute questions, please put them in the chat. If not, we're coming up on the hour, so we don't want to take too much more of Erin's time. We could learn from Erin all day because there is so much to learn, and you are such an incredible asset. I think I should say that we were neglectful at the outset. We should have introduced Debbie Olson, our executive director, who is here. She is an incredible asset, and Erin talked about her at the beginning. So Debbie, why don't you unmute yourself and say hi?
Hello, everyone. And I just want to say, Erin, thank you for your kind words. And I've enjoyed working with you for the last 20 plus years. And I'm so honored that IEF has been able to support you and the work that you've done. When you look at the advances that have been made, it's incredible. And I just hope that we can put this, in 5 or 10 years, we look at this as something that happened in the past, and we don't have to worry about EHV in our elephants any longer. So that would be a dream. And if that dream would come true, it's because of you and all the other people working so hard on EEHV for so many years.
Well, thank you. And it wouldn't have happened without IEF. You guys have just been incredible supporters of everything EHV.
Well, thank you so much. I know that I learned from you today, and like always, every time I see you speak, and thank you so much for doing what you're doing and taking your time to share with everyone. We really appreciate it.
Well, thank you for letting me.
Julie, do you have anything to wrap up?
Yes, again, thank you, thank you. Fascinating information. If you were inspired by this, of course, we'd love to see donations come in to help support this work. Also, while I have everybody on the line, our next upcoming chats are happening on May 11th. We have Dr. Karen Carter, and on June 15th, we have Northern Rangelands Trust. So, and I think we have a date set for July 31st as well, which is World Ranger Day. So that'll be a really interesting chat as well. So watch your emails and please join us for our future chats. And thank you so much for joining us today. And if you have any questions, feel free to email us directly and we'll be happy to get back with you. So thank you very much, and everybody enjoy their days.